I am very happy to be here. I'm especially happy because this is such a wide-ranging age audience. I usually talk either to students, to postdoctoral folks, to older individuals, and now I get to talk to kids as young as 3, 4. 4, okay, and another 6-year-old back there. This is a lot of fun. So, first of all, I have to say, every time I do a study, I have to say that I have no financial interests in this, so I don't. I also have no money, but my daughter is in the crowd, so I shouldn't say that, but, oh, we were doing so well, too. Okay, that works. That doesn't. Yeah. But the laser's working.
No, it's the wrong button. The arrow.
It's this one, right?
Arrow, yeah.
Okay. You know, it's like when your computer breaks and you bring it in and then it works perfectly and the guy looks at you like you're crazy. That's the same thing. So, here's what I'm going to try and talk about this today, try and get through everything. But again, it depends upon when Uroš starts waiting. But it's a brief, I'm going to talk a little bit briefly about Alzheimer's to make sure we're all kind of on the same page, and then we'll talk a lot more about it. But I'm also going to sort of walk through the early and the current landscape of Alzheimer's disease research. I'm not necessarily, I'm going to start out going by time, but you will see that once we hit the 1990s, sort of everything explodes and it's not even worth, we'll just talk about everything in general. So we'll talk about where we stand now and maybe a little bit about what we do in the Alzheimer's Center at Michigan, some of the research we do. So, first of all, the first thing is sometimes people ask me what's the difference between dementia and Alzheimer's disease. So dementia is the overarching sort of umbrella term, and it's a dementia means a decline in cognition that's significant enough to affect your everyday life, right? So that's a dementia. Now, where Alzheimer's disease comes in, you can see here, it's Alzheimer's, you know, I don't know, use the pointer, use my fingers, it's easier, but Alzheimer's disease is really 60 to 80% of cases of dementia. That's why the interest. And if you look at mixed dementia, it's most often mixed with Alzheimer's disease. So if you want to look at the, I mean, this is not to say that vascular dementia, Lewy body and frontotemporal dementia, we have separate centers for those studies in our center, but still Alzheimer's disease tends to take the biggest chunk and be the biggest concern. Good, I didn't want him to come up again. It's no problem. But I also want to, you know, when we're talking about Alzheimer's disease, we have to remember it's a continuum, right? So we have cognitive impairment, we have mild cognitive impairment. Mild cognitive impairment we know is a risk factor. About 50% of people diagnosed with mild cognitive impairment get Alzheimer's disease within 5 years. So it's a clear risk. But honestly, about 40% of them stay there, and about 10% revert back to normal cognition. But once you start this process of mild dementia, moderate, and severe, it is an ongoing process, and it gets worse. But the interest is that we know if you have mild cognitive impairment, that's a particular group of individuals of real concern because they're at such high risk. Why worry about this now? You know, why is everybody so agitated? Well, here's the reason why. If you figure that if you just take 85-year-olds, 50% of 80 people 85 years of age and up have Alzheimer's disease or have some dementia. So that it's a very significant chunk of the older population. And why do we worry? We worry because these guys, me, it's my fault. Baby boomers, you know? And you look at what happens as we go along in the years, baby boomers become more and more predominant, and 85, all of these older categories increase. So you start thinking about this, And then you look at the, these are US statistics, but the statistics in, I can show you for Europe are the same, but if you look at the numbers, these are the projected numbers of individuals with Alzheimer's disease. By the time we get up to 2060, we're close to 14 million people. It's hard to imagine that many people with this disease. So this is the big problem that's being created. And think about what it means to society, what it means to how you build buildings, because not only is it an aging society, but it's an aging society aging with cognitive difficulties. So we're doing work with architects on how better to structure buildings, how better to structure facilities for exercise, things like that. All has to be because of these changes. And it's a global change. This is affecting everybody. It's not just the US. Baby boomers were everywhere. And if you look at the number of people affected in the millions across, it's a dramatic, it's in Asia, it's a huge change. Country like China with a one-child policy, suddenly everything depends upon that one child, and if that one child isn't there, what are people going to do? So again, it's a significant aging society. In Japan especially, this is a real concern as well. So these are really significant statistics. Africa, the numbers seem not so small, but you have to remember that in Africa, many individuals in our research have a limited lifetime because of HIV. But now that with ART cocktails, people are living much longer, so they're already at a heightened risk. So that number there for Africa is an underestimate. So I was going to do a walk, but I decided to take the tram because I enjoyed taking the tram the other night. So I changed this from a walk to a tram. But anyway, our first tram stop, and I won't talk too much about this because I know that Dr. Kavcic is going to talk about Alzheimer's. But the first stop is June 14th, 1964, when Alzheimer is born. And he was a very strong student. His interest was really psychiatry. And he was an incredible observer even as a child, and even as a student. And let's face it, to do your graduate thesis on the glands that produce earwax, you have to be pretty meticulous and interested in how the body changes. But after he finished his studies, he went to hospital. He worked in the asylum in Frankfurt. He became the director there. And while he was there, he met Nissl. And Nissl was doing a lot of really interesting work on brain slides. We're talking about 1900s, early 1900s. And so here you've got the four guys: Alzheimer's, Kraepelin, and Nissl. These are the sort of key figures in psychiatry. And so the other thing you have to remember about Alzheimer's compared to the other ones is Alzheimer's was a clinical psychiatrist. He was interested in patients. So he's the one who studied patients and went out and looked. And so when he was directing the hospital, he got to Munich. He was working with Kraepelin at the university in their neuropathology and their psychiatry group. And his first study was a very large study that's still quite famous on syphilitic patients. But then in 1906, this lady that he had first met at the asylum in Frankfurt when he was directing it, he became fascinated with this lady who was there named Auguste D. And when she died, he immediately asked for the brain so he could look at her brain because he knew there was something very different about her compared to the rest of the patients. And I was telling somebody, we did a similar thing probably in 1980, 1983, 1982, when we went and took patients out of a mental hospital that clearly weren't. It's been that long that people are still doing this. But he knew there was something wrong, and he felt it wasn't schizophrenia. And then and he looked at slides of her brain. This is his slide deck. And he drew pictures of what the axons, the nice looking cells, actually looked like as they were twisted and elongated. And there were these areas of these sort of concentrations of plaque around them, which was simply not normal. So he saw both plaque, which are clumps of things that shouldn't be there, and tangles, which are cells that aren't well formed. And he wrote about this, and he then published a second case in 1907, which is actually interesting because it was slightly different. This person only had an amyloid disorder, so you only saw plaques and not tangles. But the big change came in 1910 when Kraepelin wrote in his famous psychiatry textbook, and then we have this new disorder called, that I think we should all call Alzheimer's disease, that my student found. It's been here, but he identified it, so by goodness, this is what we're going to call it. But, you know, we live in a land of politics and everything, and I should say, I'll talk a little bit more about that in a second, but let me highlight this for you. This is why he was meticulous. He wrote about her constantly as he watched her change, but he talked about her memory being impaired. She was writing and she would forget what she was doing. She was calling things milk purr rather than cup. She had anomia. She had practice problems. She does not understand questions, and she does not remember how to use certain objects. This is everything that Alzheimer's did. This is everything that Alzheimer's disease is. He named it, he identified it, and he showed the cause with these plaques and tangles in the brain. Now, the controversy was a little bit is that Oskar Fischer was doing the exact same work, but in the wrong group. He was working with Pick, and at the so the two Munich universities were sort of fighting each other over what was what, and some people say that it ought to be called Fischer disease rather than Alzheimer's disease, but Kraepelin was faster and gave it the name. And it's an unfortunate story because he was actually died in the person who was working. But he also drew pictures of these plaques and these tangles. So let's look at the second tram stop. This tram stop is in 1963. So we didn't see anything until 1963. And in 1963, Terry and Kidd are very famous for saying, if we look at microscopy, we can see these cells that look really different in some people as they age. So this is the first really pop-up of this disorder again. And then the third stop that's an important stop is Martin Roth, because Tomlinson, Roth, and Blessed really put together the first look at Alzheimer's disease as a thing, and first really said this disorder is what happens in some people as they age, not just in 51-year-olds. So that's the problem, when Alzheimer's met August Dieter, she was 51 years old and she died at 54. The same with his second case, Johann. Now, people hadn't looked at the older cases that Fischer found, but it was thought to be a disorder of the young, so it was extremely rare, and we know that she had early onset Alzheimer's. That's around 200, now some evidence suggests 300,000 people in the world have this disorder, so it's actually not very strong. So really nothing occurred until Tomlinson, Roth, and Blessed said, hey, wait a minute, this is a real disorder. And as a matter of fact, when people die, when they die, if we actually look at the plaques in their brain, it relates to how they did right before they died. So this was the first really thing that said, hey, wait a minute, plaques may have something to do, and tangles may have something to do, these abnormalities may have something to do with this disorder. And by the way, this disorder is not just in a couple of young people. So it was really a big deal. So let's go to the 70s. So in the 70s, this is another set of important things. But now by 1970, people are pretty much saying, okay, there's something different between getting old and having Alzheimer's disease. Because, really till the late 60s, 70s, oh, everybody gets old and can't remember things. It really is not, if you think about it, till this age when people really started saying, I mean, this is just a few years ago as far as I'm concerned. Of course, I forget everything with how many years was COVID, but if you think about it, this was very recently that people actually said there's some difference in this between getting old and having Alzheimer's. So the first actual sort of test, the mini-mental state exam from Polstein, McHugh, and also Breitner and other psychiatrists, really was the first measure that said you can use this test, the mini-mental state. Now we also have the MoCA, but you can use this test to classify how severely impaired somebody is. The other thing is Bob Katzman wrote this really influential editorial, The Prevalence and Malignancy of Alzheimer's Disease, in 1976, and said, whoa, wait a minute, folks. This is going to be a problem because this is affecting far more people than we think. And actually, the Alzheimer's Association was founded in the US in 1979 because of so many concerned families. So, but look, the difference is amazing. So the last thing I'll do before sort of looking at things in general is the fifth tram stop is the 1980s. And this was an exciting time because the first time Monty de Leon and Ferris presented with positron emission tomography looking at the amount of glucose used in the brain, and they're the first ones to say, look, there's a difference between controls and patients in how much energy they use. We can probably tell the difference. And as a matter of fact, if we see these changes early, it may suggest that that's what a person's going to get. The other thing is in 1984, the Alzheimer's Disease Research Center network was started. And that was in Baltimore, Boston, New York City, and San Diego were the first centers. My wife and I worked at the New York Center. But these were the first centers of purely the study of Alzheimer's disease funded by the government. Now there's 34 or so of these places now. But again, it was the beginnings of the National Institute of Health's realization too that this was a serious problem. But look, 1984, we're back to this is the very first ever clinical diagnosis of Alzheimer's. So it took us till 1984 before somebody basically simply repeated what Alzheimer's said. And said, oh, we have this disorder, it's different from normal aging, people have memory problems predominantly, mostly memory problems, and they have all these other things. So this was the first diagnosis. After all that time, now physicians had a way of diagnosing Alzheimer's. Who knows what they were doing before? But the other thing that happened is, Briand's group, a French group that's also in Brussels, they were another group that really pointed out, wait a minute, there's more than just amyloid here. We're looking at differences in tau in these tangles. So they added that second piece to the puzzle where it's not just amyloid, tau has something to do with it as well. But so the 1990s, nothing happened in here because this poor August Dieter was 50, and if you think about it, I think a lot of people just thought it's not very interesting. When Ann and I were working in New York, it was very interesting because we were waiting till people who had Alzheimer's disease, our belief in Alzheimer's disease, right before they died, rushed them into a scanner. But one of our issues was there's a lot of older people we're finding with this disease, and it's not like normal. So we weren't finding a lot of 50 and 60 years old. We were finding a lot of people in their 70s and 80s, and it made a big difference. And you can see all these different changes that I'll talk about in a minute that come along. And this just as an example, this is the Rochester pediatrology group in Minnesota and Wisconsin. This is the change in diagnosis of a the diagnosis of Alzheimer's was entered in 1975 in this group, but really it took till the 2000s before there were patients being diagnosed with Alzheimer's. These are the hundreds of people diagnosed with Alzheimer's disease back then. And look, you're running into 2012 before you're really seeing a lot of individuals who are actually diagnosed with this disorder. So it's sort of been this hidden disorder all this time. Now, so the other thing that happens then in the 90s is the first attempt to actually say not just a description of the symptoms, but now we have enough information to say something about the mechanics. Now, I'm going to show you this in a simple form rather than with a million little arrows going back and forth, because everybody argues about which came first, tau or amyloid and tau, whatever. But we have a mechanism for this that suggests that you get an increase in the brain of amyloid because precursor protein, which we all have, which enzymes break down essentially to feed our brain, the wrong enzymes get in there. It's like the bad cook gets to the hotel and the food is terrible. And what happens is you get amyloid deposition. You get these clumps of amyloid that didn't get cut up right that begin to blockade, essentially form blockades in the brain. And then that leads to inflammation because the brain is going, what the heck is going on? And it's beginning to, white cells, mitochondria, everything's going nuts trying to take care of it. So you see this inflammation, and then you see tangles beginning to form, because what happens is the axons, the messenger parts of these cells, begin to fall apart. What these axons are made of is tau. Tau is the cement that keeps them together, and now tau begins to fall out. So what you have is clumps of amyloid that doesn't get rushed out of the brain because, frankly, the brain can't get rid of a big clump. And you have tau that's falling out rapidly out of the cells. It itself is then running back into the cells, and that's what causes the tangles. But you get this sort of cascading effect, and then the cells themselves begin to die. But this is the time when we diagnose this disorder. Way too late in time. And all this stuff has gone on probably 20, 25 years earlier. Because we know that amyloid enters the brain very early on. And we know that amyloid appears 20, 25 years before. So now we're stuck with this problem, but this is the only place where we're treating people. And we're treating them basically with drugs that sort of add a little choline to the system, and maybe the person can think a little brighter for a while, but it's not stopping cell death at all. So what you get is, rather than a normal, nice, neat brain full of axons from neurons connecting to each other, you get these bundles of plaque that bump into cells that don't clarify. You get the cells themselves beginning to fall apart, forming these tangles. And that's Alzheimer's. Alzheimer's saw that in 1906. The rest of us didn't pick it up till about the 70s or 80s. It's a really interesting difference. The other sixth tram stop, so my other sort of important stop now in the 90s and 2000s, is the ability to look at imaging of the brain. This changed everything. The beginning, Monte de Leon, Kurtz and others really brought in this whole idea of we can now image the brain. So I'll show you some basic images. This is what I grew up on. I mean, you all get to do fMRI and imaging, all sorts of things. I grew up with PET scans. We had to have the person sitting there for 45 minutes, an hour. It was a very complicated process. But what it showed us first is that a person with Alzheimer's disease, if you see the more red, the more energy used. Because this uses basically fluorodeoxyglucose, basically imaged sugar. So you can see in the right-hand side, the normal individual who's 71, there's a lot of activity in there. Even in the occipital area, because they had eyes open. in the temporal, parietal, even in the frontal areas, but that's the patient with Alzheimer's. Parietal, temporal, especially areas, are just not there. So we know this itself predisposes this, which is the actual MRI changes, where you get cell loss, you get atrophy, you get widening here, and you get loss within the critical areas of the hippocampus. They begin to die. This is when cells die. This is the process of when cells are beginning to die. This is the process of when those cells are dead. Now, when you want it to move, it moves. And now I touch this button. Sorry. Don't come back up and find me. I got control. So, I can be careful about what I put. And also, Uroš reminded me, don't push that button. You know, and there I go. But this is the biggest change in Alzheimer's, because now we can image amyloid. And we've been doing it for a while. We can actually now, in this case, the brighter and the more red, the more amyloid in the brain. So this is a patient with Alzheimer's disease, and you can see amyloid in the frontal areas and the temporal parietal areas. In a healthy control, maybe a few little spots here and there, because we all have a few little spots. But there's a dramatic difference. So now we can measure plaque in the brain. Now what does that mean? This means this is that thing that happens 20 years before. So it's very exciting. Now we can do something else. We can now measure tau, the tangle, the things that come afterwards that forms tangles. We can now look at the brain and we can see differences. We can see that you get a lot more amyloid in sort of the frontal areas and upper areas, cortical areas of the brain, whereas tau seems to be much more centralized towards the hippocampus. But somehow it's an interaction of these two things that causes Alzheimer's disease. And we'll talk a little bit more about that in a minute. But imaging is a unique key for all of us. No, I didn't do it yet. There we go. So let me go back one and I'll just show you the difference. This is what we know happens. We know that there is in people who are developing Alzheimer's disease, there is this very early presentation of amyloid. Remember I told you that. About 20, 25 years before, that amyloid has already hit its peak before somebody is even diagnosed often. And then following that, you get this, which is the appearance of these other things that match the disease much more closely. You get images of, you get tau markers, you get fluorodeoxyglucose markers, cognitions, and eventually, further down the part towards dementia, you begin to get a severe change in cognition. But we know that amyloid is an early marker, and we know that tau, those tangles, those are the sign that something is really happening. And we also know that this correlates dramatically with FDG glucose and with cognition. These things correlate together, but we know this amyloid has already done its damage. 7th tram stop. So our first diagnosis was in 1984. So then it took till, and that diagnosis was all about Alzheimer's disease. And in 2011 was the first change in that. The same group of the Alzheimer's Association, National Institute of Aging, and a whole group of individuals got together and looked at that diagnosis, and for the first time said, you know, we're looking at all this imaging, and we're going to call this, for some people, preclinical Alzheimer's disease. So we're going to call this no apparent symptoms, but there's an amyloid buildup. some people have said that this really was a result of drug companies wanting to make more money to treat people with amyloid, but it's true. This is the first marker of things to come. And that criteria also talked about very carefully about mild cognitive impairment. Problems that are greater than normal, but it called for two things: better diagnostic tests and measures, and it also called for more research to specifically be able to use measures of CSF or measures of imaging. Now, what's the difference between CSF and imaging, right? The difference is, for me to get a patient scanned with amyloid and tau costs me about $6,800. For me to get amyloid and tau out of the CSF costs me $450. For me to get a volunteer to do a spinal tap for CSF in the US is not a chance in the world. So again, different areas, you know, I feel like a friend of mine sort of talks like the Dutch, if they have a headache, they get a CSF, they do CSF. But in our country, it's very hard, which is too bad. Because what does the CSF look like? What the spinal fluid looks like is a lot of tau, because the tau is falling out, and very little amyloid, because it's up there clumped and it can't get out. So you can diagnose as effectively with CSF as you can with amyloid. But then, that was in 2011. Now, in 2016, Clifford Jack, who does a lot of this work, who did the first one, he and a group, you know, Clifford is an imager at heart, and they said basically, we suggest there should be a reclassification of Alzheimer's. And this is where we sit right now, that we're going to call Alzheimer's disease an unbiased approach. So I don't care really what the person looks like. I don't care what the behavior is. I don't even care which thing came first. But we're going to say it's a biomarker category. So we're going to do a breakdown. And we're going to say if you have no amyloid, no tau, and no neuropathological changes in the brain, no changes on MRI, we're going to call you normal. I don't care what you're doing, we're still going to call you normal. We're going to call somebody who has amyloid, but without tau yet or brain changes evident, we're still going to call that person has Alzheimer's pathological change. So we're essentially going to call that person Alzheimer's. And then, if we run into these sort of things, that becomes Alzheimer's disease. And then we even have categories if there's no amyloid, but if there's tau, we can look at all these different mixtures of these other disorders. But the key to this is, and the thing that's causing so much fury is, you know, Clifford goes, you know, hey, this is just the way it is, and everybody else is yelling, but where's memory, where's everything else? And this system has an effect, and something I'll talk about when we talk about treatment trials. But what's the suggestion is, I don't care what the behavior is, I can measure it with CSF, or I can measure it with PET, amyloid, and tau imaging, and I can tell you whether that person has a disease, regardless of what their behavior is. It's a pretty dramatic step, and it's causing, people are still irritated over it. But people are kind of coming along to it, so same thing. So Jack came up with another model. Remember that other swishy model? Now he said, rather than looking at illness, you know, normal, mild cognitive impairment, and so forth, we're just going to look at time, and we're going to see where the detection threshold is in the abnormalities along this way. Where do we really see a amyloid disruption, and then look at the others, and we can sort of do diagnosis that way. So this whole idea of early diagnosis in order to treat has become a big deal. We've already talked about You know, the temptation to hit the wrong button is just intense. There we go. Whoop. So, we've already talked some about imaging and how important that is. We are doing a lot of research right now looking at vision, because amyloid does get deposited apparently in the eye from the brain, so there's a lot of interest in that. And then this is the sucker, that the guy in the middle is the sucker that's got everybody, right? Because you go to your doctor, you get a blood test, oh, your creatinine looks like this and your bilirubin looks like that or whatever. Well, what if they said in your risk of Alzheimer's disease is the following? So everybody and their mother is trying to get a drug. So the first, this is just I mentioned this real quickly. This is the IDEA study, because one of the things is PET scanning is expensive. And so the Alzheimer's Association and CMS, the medical unit in the United States, did a big study of Alzheimer's disease, and now they really find that it is worth paying for scans, because if you look at that, 2/3 of people who got an amyloid scan had either a change of diagnosis or a change in treatment. So if you have what looks like Alzheimer's and you get an amyloid scan and you don't have amyloid in the brain, I shouldn't be giving you anti-amyloid drugs. So there's a lot of proof now that using that test, because remember, if you have Alzheimer's disease, you have to have amyloid. That's the key for all of this. You also have tau and other things fall apart, but you have amyloid according to that criteria, then you have Alzheimer's. But the blood test is the big thing. And I think it was a couple years ago at Alzheimer's International Conference, they were talking about pretty soon we'll have PET scanners across the world doing amyloid and tau scans. Well, that's a pretty good idea, but you know, the cost of that just wasn't going to work. But if you had a blood test, it would be amazing. So if you look on the literature, everybody's got a different test. This is P1, this is P217, there's P-tau127, there's a lot of these, but P-tau 2017 is interesting because it really does appear to differentiate Alzheimer's from other types of dementia. It really does pick up both on amyloid and tau. It appears to relate to the stage of the disease. It identifies appropriate people for clinical trials. That is, now it could be used as a way to say, okay, we're going to look for people who clearly have an amyloid problem, and it looks like it starts to appear long before symptoms are caused. So this is the first of those tests along the line. But I can tell you there's a million of them out there because everybody wants to have their tests. So let's talk about clinical trials for a minute, because this is where stuff has gotten interesting. So I just want to mention to you first, you know, you think about a clinical trial, and it's really, this is the zone where we want to intervene, right? Of course I'm concerned about people with mild, moderate to severe dementia, but if I could stop it, either as an MCI or pathology, that would be the key. And what type of stop is it? Is it a cure, so the person is normal again? Do you just arrest the progression? Right now, this is what we do. We treat the symptoms. This is all we do right now. So this is just to give you that timeline again. Here comes Alzheimer's in 1906. Around 1976, people start saying, hey, wait a minute, you know, that guy was talking about stuff. This seems to be really happening everywhere. And then it took from 76 to 93 before the first drug appeared, Tacro or Cognex. And this, when we were in New York, was sort of the hot topic. Originally, you had to take this every 45 minutes. So it wasn't really that effective as a measure to use, but still it was interesting. Now we have a whole bunch of other things. Namenda works on the glutamate system, but the rest of these are essentially flooding the brain with acetylcholine in order to improve transmissions across neurons. But all they're doing is helping symptoms. So the excitement this year was this one. this was potentially the first drug to actually modify the disease, right? So this would be the real game changer. If you could go in and get rid of amyloid and maybe even affect tau, you'd be in great shape. So I didn't do that. It's hot. Oop. Okay. Very briefly, this is what drug studies are like. Phase 1 usually includes 10 people, usually younger, healthy individuals. And the question is, let's see what happens if I give them this drug. Now, they're usually nice college students at a university like University of Texas, you know, and we have a phase 1 trial. And here you want to study, you're doing your prelims or you're writing your dissertation. Come on, you know, you have to stay in bed flat, but we'll fix you up with a nice computer, and then we'll kind of give you the stuff and see what happens. That's phase 1. Phase 2 trials are usually around 100 patients now. Patients with Alzheimer's disease, and now we're beginning to say, okay, we didn't get rid of these guys up here. It didn't seem to affect them or hurt them. Now we're looking at whether this drug is actually beneficial. And then phase 3 are what we're used to, very large trials, usually a matter of years rather than months, to really look at the effectiveness. Because that's the key. Babenuzumab is a good example of that. Babenuzumab was a drug that actually had been an allergy drug in Russia. And everybody got excited because older individuals who seemingly took this drug didn't get Alzheimer's disease. So that got, supposedly, so this got there was $1 billion spent, basically, to develop this drug and market it and run it through the studies. And once you got down here to the phase 3 trials, it was a complete failure. It's the reason why Pfizer now won't do studies in neurology. They just said, we just wasted a fortune on this. We're not going to do it. So these studies are incredibly expensive to do, but they're what's required to show proof. And usually you have to do 2 of them. I'm sorry you're recording this, and the guy's going to be going boop and all sorts of things all the time. But this is aducanumab. This was the first study in Nature with Biogen's aducanumab. And let me tell you, I was at the AAIC meeting before this came out, and the nice professor giving the talk about it said, now please, no one take a picture of these slides. And I'll tell you, every single telephone went up in the air to take a picture. But look at this effect. Again, we're back to small studies versus big studies. This only had 165 people with mild cognitive impairment. But look at baseline to 1 year on placebo, 3 milligrams, 6 milligrams, and look at 10 milligrams. This is the amount of amyloid in the brain, which is essentially practically normal. This is the patient when they came through the door. This was exciting, but remember, we're talking about 165 people divided by 4. It's not exactly a big group of people, but it was enough for Biogen to say, we think we have a signal here. And everybody at the Alzheimer's meeting to kind of applaud or go, whoa, or let me go rush and put money into Biogen. You know, it was an exciting time. The reason why I want to show you this is I also want to show you what it takes to get a drug into use. So Biogen did 2 things. It had 2 studies, because remember, you have to do 2 phase 3s. So it had a study called Emerge, and it had a study called Engage. And both of those studies were stopped early because of side effects, because of what they call ARIA. I'll show you in a minute. But they both stopped. But there was a signal here, 22% less decline in what's called CDR, the sum of boxes. This is essentially a clinician's rating of how much impairment this has. So on the highest dose, out around 78 weeks, they were beginning to see a signal. So this got to be really of interest, but Engage hadn't changed. So how come you can have 2 different studies with 2 different effects. And yet they're both exactly the same design. Anybody know the answer? It's actually a very simple answer if I could get you there. There. Both studies look like this. They had ApoE4 carriers, which is another one of these genetic risks. And the idea was, oh, we're not going to give them as high enough because maybe ApoE will get interacted. And in the titrated high dose, 6 milligrams and 10 milligrams. And what happened in the middle of the study was there was no signal here on an early analysis, but there looked like there was a signal down here, so everybody was switched to the high dose. The problem was one study had started a number of months before the other one, so you didn't get as many people with the high dose. So what they would like you to, Biogen would like you to believe is both studies would have been effective if we'd only started them at high dose at the same time. That's called protocol deviation. So, but the other thing about this drug is it has a very high content of side effects. 40% of the people had side effects. Granted, the statement was if you waited 4 to 16 weeks, that would go by, but as a miraculous drug, there are big questions involved. So, however, Biogen said, look, there's really something here. I want to push this. We had to stop these studies, but we've begun following the people who wanted to stay on drug, and we're seeing differences. We're going to take this drug to be approved. So what happens next is you do your 2 trials, even though Biogen never really finished either of those trials, and you go to the FDA, the Federal Drug Administration, and they bring in a group of knowledgeable scientists, you know, like with beards and stuff and canes and things, and they say, is this drug effective or not? Is this drug, what do you guys think? So there were 2 questions. One was, does the second study that showed a difference, is that enough for us to approve this drug? And one person did say yes, but 8 of them said no. And then further, if we look at those 2 studies along with that really pretty slides I showed you the first time, now would you approve that drug? And 10 of them said no. So science said no. These studies were not completed. They're protocol violations. We don't want to do it. Much to everybody's surprise, however, the Federal Drug Administration said, well, you know, wait a minute. There's a lot of people out there who have Alzheimer's. There's no drug out there. We want some more information. And Biogen gave them more information in the study. And for example, in amyloid PET, if you only look at high-dose people in both of them, amyloid changed significantly. Even the sum of boxes, even the sum of boxes changed significantly for both of them. So Biogen's claim was, look, there's a real signal here. We want to get this drug. Again, you have all the scientists who said no. And they also said, you know what? If you look at measures of tau, we're even seeing some differences between tau over the year. The tau scans also, not just amyloid, but we're showing a signal in tau. Oh, come on. Wait. I know Uroš is sitting there squirming. So the FDA said, you know what? We are going to approve this drug. Because we're going to say that this is an emergency because there isn't another drug. You scientists keep telling us amyloid is the key, and we know there's a signal with amyloid. So I don't care if we didn't see behavior. We're going to approve this drug, and we're going to approve this drug just because it changes amyloid. And, you know, scientists went nuts. You're taking a drug with high side effects. You're doing, but FDA is FDA. So there are a lot of concerns. At last year's AAIC, Alzheimer's International Conference, which, by the way, is the only conference for just one disorder really in the world. So at the AAIC conference, there was a big panel that argued back and forth, and they basically said, look, no more severe patients, only mild cognitive impairment and mild Alzheimer's disease should be taking this drug. Highest dose should be used only, and you've got to give people MRIs to monitor for ARIA. Michigan said, no thank you. The VA said no thank you in Michigan. Wayne State said no thank you. Everybody said no thank you. We're not going to allow this drug to be administered here because we're too worried about the side effects and the amount of cost it'll take to do it. Now, of course, I can tell you that all sorts of little infusion sites, because this is an infusion drug, all sorts of the infusion sites that are doing infusion for other things suddenly said, but we can do it. You know, so that caused a lot of stress. Adanucanab, by the way, just was disavowed in Europe as well. You won't see this drug here. So think about all the money that was spent on that. But the final straw in this problem was the Centers for Medicare and Medicaid, CMS. They said, you know, FDA is interested in new drugs and helping problems and trying to get things out to market. We're worried about individual people. We're also worried about our pocketbook. This drug was running first for $80,000 and when everybody a year, and when everybody complained, they said, well, we'll give it to CMS for $40,000. And CMS said, no, thank you. Because CMS said, you had a study, you did 2 phase 3 trials, they didn't finish, your endpoint was supposed to be behavior, and FDA said it's okay not to do that, but we're going to say you have to find that behavioral change. But they also said, importantly enough, they said, if you want to take this drug, the only way we'll give it to you is part of a university trial. We're not going to pay for it if it's some little infusion site, but we're going to apply the same restrictions to every other study coming down the pipe for amyloid, that we want to see 2 trials and we want to see change before we'll pay for this drug for people. And so that's where we sit. We still have drugs moving through. About 62 of them are actually in phase 3 for different types of dementia. But the most important thing about aducanumab is it may have failed in marketing, and I think Biogen will pull it off. We'll pull it off market pretty soon, but I'm not sure about that. But it just never got traction for cost and everything like that. But one of the other pieces on this was it went first, and you needed somebody to go first. And now these other drugs coming down the pipe, Biogen's BAN2401, Eli Lilly's drug, and Genentech's drugs, these drugs are moving along, but now they've seen, the first thing they said when Biogen got approved, they said, hey, we've done phase 2 trials, approve us too on an emergency basis. But now nobody's doing that. Now they're going back, they're completing phase 3 trials, and they're looking. There was a lot of interest in Roche's drug, crizanlizumab, and they just announced June 16th that it didn't have an effect. There was no slowing of Alzheimer's disease in persons with high-risk genetic linkages. So from a drug standpoint, it doesn't look so good. So we're back to the point. Maybe amyloid isn't the right target. We all agreed it hits its height. Maybe that's the wrong target. Maybe we should look at other things. So there's a lot of new drugs coming down the pipe. We still haven't found a workable tau drug because we know tau correlates best with memory impairment because tau comes along long after amyloid and starts to lift and clearly predicts memory impairment and things, but no tau drugs have been effective. So we're stuck with the problem of we know that's what's going on up there, and the chemistry of it should be pretty clear, but nothing works. By the way, if I take a mouse bred for amyloid, and even the amyloid tau mouse that's now available, you give them this stuff, it clears right away. So we can solve a mouse. We can't solve the human brain yet. Humans are just kind of messy compared to mice, but it's an interesting feature. So my final stop is Giuseppe Tartini Square, and there's Giuseppe Tartini and his famous The Devil. You know, there's an American song about Devil Went Down to Georgia, and there's Tartini had his dream about playing against the devil sort of thing. But it's true, you know, play music, it was actually a help. He was actually helping himself. But the whole point of this is, okay, we don't have a drug yet, but what can we do to make a change here? What can we, if we could just delay by 5 years, Voyko, we have a meeting right now. If we could just delay by 5 years the presentation of this drug, by 2050, close to 6 million people wouldn't develop Alzheimer's disease. So rather than this massive hit coming down the pipe, we have a change. You know, that's just, can you click that? Otherwise, this is going to stay here forever, and then they're all going to fire off up there. But that's the whole point. What can you do? Oh, that's right. Never mind. So what can you do to affect this? Now I'm going to get a text that says, "Where are you?" That's the next question. So the whole argument comes down to, we don't have a drug. What can we do as individuals? Can we do anything? So where this all changes is all the studies that have been coming, frankly, mostly out of Europe. Americans started this route a little bit too slowly, but looking at what are the risk factors for this disease. What are the risk factors we can change and the risk factors we can't? So I can't change age. It's about 55% of the risk. I can't change your ApoE genotype. For the first degree relative, that's about a 5% risk. And I can't change the fact that more women get this disease than men, even considering the fact that they live longer. But there are a lot of other risk factors that are modifiable, right? We'll talk about some, but there's the more educated, the less risk. Hypertension, obesity, all of these things fit together. And the proof of it was in the, let's see if in the Lancet study. So the Lancet, 2 years ago at AAIC, did a huge review of all the literature on risk factors and came out with the fact that all of these things are contributing to make up all the rest for age. Now, this was the one that surprised everybody, that it has an even bigger risk than ApoE, was hearing loss. But all of these things along the way, and all of them at different parts of the lifestyle in particular. So that's why lifestyle has become so interesting, because we don't have a drug yet, but if we could reduce our risk by changing how we live, that may be the key. And these big studies show we can potentially do that. So I wanted to mention this about women because it's interesting, and it shows you the complexity of this problem. Women have certain different specific genetic differences that appear to describe risk and resilience different than men. Women have a faster rate of memory decline actually among those who never joined the paid labor force. And the tau networks appear to be differently distributed in women than men. So you have even a gender difference, let alone differences across different populations. So all of this now makes this whole process different and more complicated. But we know that those risk factors sort of override. This is the big one too, of course. This is the American diet I aspire to, is fried chicken and a fry burger, which actually is sold in the US. But this is dramatically the wrong diet. And why is it the wrong diet? Because this diet leads to hypertension, leads to diabetes, leads to all these medical problems that all contribute to Alzheimer's. The MIND diet is sort of taking the US by storm based on the Mediterranean diet and the salt-related DASH diet. but it has an effect. If you look at individuals on the Mediterranean diet, they really do show a significant difference in their risk. Hearing and vision is another one. I swear I'm not touching it. Hearing and vision is a big thing. Hearing caught everybody by surprise, and I can tell you the visual guys got really upset and said, what about us too? But we do know that if you have a vision or a hearing chance, your risk is increased by 10%. If you have both, it's increased by 112%. Why? And other studies that have looked at sensory difficulties, changes in touch, changes in smell, changes in sensory functions, all of these things increase the risk. Now, you could say, oh, it's because somebody who has vision and hearing doesn't leave the house, they become depressed. That's part of it, but that doesn't seem to be all of it. So in Michigan, we've got a whole group of individuals looking at hearing, including looking at rock musicians who tend to lose their hearing and tend to act a little strange. Sleep habits. We know sleep is important. It's another thing that we can all do. The better we sleep, the less amyloid gets deposited, because the amyloid, at least what can get flushed out, gets flushed out at night while we sleep. Mental health care, we know depression from the Faenza study in Italy and other studies have shown clearly that stress and mood has an effect. Mental activity. This one is a brand new study that's really quite interesting because, you know, we've all been saying keep your brain active and you know, it reduces the risk and things. So Wilson's study, they looked at a very large sample of people and looked at how many of them had high mental activity and how many of them had low mental activity. They didn't play cards with their friends, they didn't do puzzles, they just sat home like Homer Simpson and watched TV. If you looked at those two groups of people, there was a clear difference at when they developed dementia. That was really neat. But then the second part was the surprise, because 695 of those individuals died, and there was no association between mental activity and either tau or amyloid markers. So the interesting thing about this is somehow this stuff may be stopping an underlying process, but it's not changing the overall rush of amyloid and tau. So it's slowing that, but that's what we want, is a slowing. So I put this here so Uroš could see what happens if I try and get on a treadmill. But we know, obviously, exercise makes a difference. There's increased fitness has an increase in hippocampal volume has been shown across it. These older studies that have looked at a very selective effect with exercise to hippocampal change, and hippocampus being the center of where Alzheimer's comes. You know, and we get these recommendations from the Alzheimer's Association and from Alzheimer's International about it, but studies that just have come out this past year show that 8,000 steps instead of 4,000 is associated with a 51% lowering risk of death. Staying active has a significant effect on it. Staying both cognitively active and physically active. And why does that happen? You know, we know that there's clearly indirect effects. If you exercise, it reduces your obesity and your vascular risk and your inflammation and so on. But we also know from animal studies there are clear brain differences from those who are more active and those who aren't. And even from this is an older study, but some of the studies from mice research show that those that exercise get a proliferation of new cells, especially in the hippocampus, and then that fires up BDNF to protect those new cells. That itself is a clear protein involved in memory, and you get this real pattern of health here. You get hormones that come loose to try to stimulate blood flow, and one of the studies done in Michigan has showed even an amyloid reduction in those mice. So it clearly all fits. Physical exercise is critical. And now if you look at some of the new studies that are finally being done, who say let's look at a bunch of things at once, and then we'll decide the effect. If you combine 4 or 5 things from this list up here, you get a 59% lower risk, 39% even with 2 or 3. So combining these things we do in life and changing our lifestyle really does lower our risk. And how many of you are familiar with the finger trials in Finland? So this was a hot topic, right? You know, they showed up at Alzheimer's International Conference a couple of years back with these results, and everybody was sort of blown away because what they did in Finland, and everybody knows that they stay attuned to what they're supposed to do, nutrition, physical exercise, cognitive training with the computer, and monitoring vascular risk, after only 2 years in this study, already showed significant differences. And this started a whole wave of finger trials across the world. And they're centered by the Alzheimer's Diseases Association's worldwide fingers trials, but there's finger trials going on across the world, all of them trying to adapt these 4 things, which we should have been worrying about right from the beginning, rather than saying, well, let's see, does diet work, does this work? Looking at all 4 of these things together and trying to see what works best, and associating each one with each country. So each country gets a slightly different version. You know, that in looking at the Mediterranean diet, it's clearly risk reducing for Alzheimer's disease. So everybody in the US started taking it apart, looking at DHA, looking at this, that. And, you know, if you simply ate more fish, you got better. It's, you know, do we really care why? It's a question. You know, we want to know that as scientists, but the first question should be, can we affect it? So this is the US study that, of course, in the US, it's I'll do it myself, don't tell me what to do. So there's two sort of trials going on. But it's really interesting work. And do I have time just to talk briefly about COVID? Am I doing okay? Okay. You haven't rushed the stage yet. I'm sorry it's hot. But let's talk briefly about COVID, because most people with dementia are 65 and older, which puts them at a really high risk of Alzheimer's. We know that there were 42,000 more deaths in 2020, a 16% increase in Alzheimer's deaths at the time of COVID. We also know that most people with Alzheimer's disease also have other underlying health conditions. So COVID is really an issue. A couple of the studies that came out in 2019, right at the beginning of COVID, have really formed the basis of other studies. You know, an Argentinian study that showed significant long-term effects. The NYU study of 310 individuals showed clear changes in amyloid and tau blood-based markers. And the other thing is, of course, we're talking about it today, all these individuals who stayed in their senior centers locked up, or in their house locked up, with looking only at their family through the window, not being able to touch people, this itself, in loneliness and depression, we know that contributes as well. So COVID is a real problem. One of the larger US studies that just came out analyzed health records from 62 million people. First of all, found that African Americans have a higher risk. And second of all, that risk was 2 times higher for people in the study with dementia. So clearly there's a risk with dementia. And we know from some of the other research showing EEG changes in those individuals who've had COVID as well. So it's a really interesting, it will be a significant issue going on. So we know it affects the brain. We're still not clear why, but there's a lot of research going on. There's a big study going on across the globe now that the Alzheimer's Association is doing, looking at the associated risks of COVID with other medical problems. Okay, this is the end, I promise. I'll tell you just a little bit quickly about what we are. I'm the co-director of the Michigan Alzheimer's Disease Research Center. We follow both Wayne State, University of Michigan, and Michigan State. These little jerks get 2 centers. But, you know, I don't like talking to them during football season, but otherwise I'll talk to them anytime. but we are a joint center. Everybody serves in the different groups and things like that. We have lots of studies ongoing. Our center looks like all the other Alzheimer's centers now by mandate. We have a clinical core that does all of the evaluations. We have an administrative core that gets to kind of make sure people do their job. We have a neuropathology core. We have a data core that does all of our data analysis. We have a biomarker core that's up actually at Michigan State, which is really doing all of our blood work and does also the blood work for me from my Nigerian study. We have an imaging core that does all of the imaging work, and that one's located at U of M. And we have an outreach and recruitment core that finds participants for studies and goes out and talks about it. And all of it is in the research education component. The rec core that's an integral part of this, looking for junior investigators, and one of them is here. Dr. Caro over there is Katie Caro is one of the rec mentees. So our rec mentees get around. So we have a lot of different studies ongoing. Real quickly, every center has to have a clinical core that monitors individuals over time. These are people who start with clinical concerns, no clinical concerns, mild cognitive impairment, and they're followed over time looking for change. And they get yearly medical, MRIs, blood work, and so forth. We have a lot of other research studies. I do a lot of, I was blessed with a very large NIH grant to look at driving. Because what is it in life? You know, we were talking about this before. What's the most complex thing you do when you're older? And I noticed this in my father in his 90s, and I went, boy, this is something I really ought to look at from a research standpoint. You get a 90-year-old driving in the car, you know, with some mild cognitive concerns, and it makes you really, really wonder what's going on. So we look at driving both from simulators, we look at driving from a fixed course through Ann Arbor, and then we also look at naturalistic driving, where we can put a little box you can't see under the seat there, and then we get camera views forward, backward, we get GPS, we get everything in the car, so we can look at how people react and respond, right? Look, and it's really important. I was talking about part of what happens in the hippocampus, and Dr. Kavcic can tell you, has a lot to do with motion perception, right? Where do Alzheimer's patients often have problems? It's trying to enter freeways. What's what you do? You look at your side view mirror and you say, that truck is coming really fast and he's going to hit me if I go out. But what if you can't really do that? You go out anyway. You know, my mother had an accident like that and couldn't explain what happened to me. You know, and that was about when we sort of slowed down on the driving. But again, it's the biggest thing we do, which requires the most attention, immediate attention, problem solving and everything. So that's why we're outfitting cars, and we've done studies with Ford and Toyota doing the same thing. It started because I used to do a lot of mobility work looking at movement and differences in Alzheimer's patients and their ability to meet and cross obstacles and things. And I started thinking, oh my gosh, what if I'm driving a car? How do I do that? So we got really interested in driving. Voyko's doing a very interesting study in Detroit where he's looking at early EEG markers for cognitive impairment. Could we find early markers that could be used in the community that might tell us who we need to look at a lot more carefully? And can we maybe even join that with some cognitive testing? We also look at brain stimulation, TDCS. This is becoming a hot topic in the US, where just about, an 8-volt or 2 8-volts worth of current pass through the brain either in small or pads, appears to enhance cognitive training. It's been used for motor problems. TDCS has been used for a lot of things, but we've got a large group really interested in looking at this for cognition. I got this in here. This is Ben Hampstead in our group who does this, now has a system that he can send home with the person. So I'm going to send you home with the TDCS unit, and you can practice these cognitive things, and then come back and see me 3 months later, and we'll see how you're doing. By the way, don't go out and buy a TDCS unit from Amazon. They do sell them, but I don't recommend you buy them. But we have lots of other studies. A lot of studies look at cognitive testing. We have a lot of studies that look at everyday health, and a lot of our trainees are doing this research. We have the REVEAL scan studies that are looking at the importance of how, and it's really important, how do you tell somebody they have a positive amyloid scan? You know, the ADNI studies, the original Alzheimer's disease neuroimaging institute, they won't tell you anything. But that's not what people want now. They say, I'll do your test, but you have to give me information back. So how do you explain this to people along the way? So there's a lot of interest in that. We have a Lewy body disease program that we have that's very large in our group. We have a large brain donation. If you want a slide, I'll be happy to arrange sending you some slides. And we also do a lot more. This is Edna there in the middle. We have programs for caregivers to get them a day off. We have studies we do within the community. We have a big wellness initiative because we're blessed with a really beautiful botanical garden nearby, so there's lots of things. And you can't see her, but I always end my talk with a picture of Laura down here because she's always doing meditation things with me. So now, if I just look at her picture, I get relaxed. So I can't see her picture, but I'm not relaxed. So thank you. I'm not touching it. I'm sorry that took a long time. It was fun.
Thank you, Bruno, so much for providing us so interesting and important lecture. I'm really glad that you came here. We managed finally to get you to Europe.
Before there's something awful on my email.
No worries. We can switch it up. So I'm a bit worried now about what the other guys from the catering prepared. So it's not Mediterranean, it's not good, so we can.
Is it fried?
I'm not sure. Okay, so the question, I would like to open this floor for questions from the audience, but before doing that, perhaps like we always question ourselves, like what are those, and you gave us lots of home messages or messages to take home, but is there, like, I remember when I was spending some time in your institution, there are medical doctors who care only about sleeping deprivation, and so forth. So then I remember there was one study coming out showing that basically our brain shrinks during the night. So it's really important to have like this non-interrupted sleeping behavior. So perhaps that could be like one of the.
Yeah, I mean, I was talking with you about CPAPs.
Yeah.
So I mean, how many people in this audience use a CPAP? Ooh. If I asked this in the United States, at least 25% of you would raise your hands.
Sleeping machine.
So these are machines that do alternate pressure while you sleep. So the people who snore or people who have apneas and don't even know it, it takes care of that and it improves oxygen flow. But that's not something that I find people do in Europe at all. I've used one for years, and so has my wife, and I frankly feel better. She used one because she really felt, I'm sorry I'm talking about you, she's probably gone now anyway, I don't have to worry about it. But she used it because she was so sleepy during the day because she had apnea, and the machine really changed her whole way, except when the cat sits on her lap in front of the TV and then you're gone. We have a cat that can put you to sleep regardless of what time of day. But the point is, I had no problem staying awake, but I have very massive sleep apnea. And so it really is something that gets treated. So that's where the sleep doctors come in. But you know, it's all of these different things. And you know, I tried to show you a lot of stuff that just when you see something else, it'll ring true in the future and go, I remember that guy talked about that. But sleep is a critical thing, and it's interesting that I don't see that here at all.
Yeah, but then what about how many hours of
How many hours of sleep is a big discussion. Usually the word is 7 to 8. If somebody's getting 9 hours of sleep at night, that's usually something funny. Some people, you know, my mother did develop Alzheimer's. Well, actually, she developed something called late, not Alzheimer's, but you know, she never slept. You know, I can remember as a child her up at 3, 4 in the morning working, and I hate to admit that I have. Is my wife in the? You know, some people don't seem to need to sleep, but they really do. I really need to get sleep because at night is when you clear stuff from your brain. So it is important to sleep. Aside from everything else, keeps me from bumping into the wall if I get enough sleep. But clearly, sleep is a key issue on this. You know, if you had to tell me what I thought was key, I really think probably number one is diet. Diet clearly makes a difference, and it's dramatic. And number two is exercise. You know, because if you take care of diet and exercise, it pretty much takes care of all those other things coming your way, like diabetes or cardiovascular disease, or at least it slows them down, which are all risks for Alzheimer's. So, you know, I laud the work you all do in motor functioning because that really is a critical feature of this.
Thank you.
Yeah.
Any questions from the audience?
Everybody's hungry.
Everybody together.
Everybody's hungry.
Yeah. No?
Yes.
Uh-oh.
Thank you. Thank you. This is a huge lecture. Maybe just a simple question for the simple problem. If you can define the ratio, the proportion between the cause of the Alzheimer's disease between genetics and lifestyle, what do you say?
I'd probably give genetics around 7% and lifestyle around 20%. I mean, lifestyle makes a huge difference, and genetics, except for, say, presenilin gene mutations and things that some people have that drives Alzheimer's at an early age. But really, it really is lifestyle. Stay in school, don't leave school. Stay active mentally. Those things really are effective, and when, you know, that Lancet list is full of all those things. Take care of your health, take care of your hearing. You know, I still don't understand hearing, that would have the highest risk of all the lifestyle things is hearing has the highest risk, people with uncorrected hearing. I can't tell you why exactly, but it really is true. Lifestyle is a much bigger chunk. Age is 55% of the risk. You add another 5 to 7% for genetics, but then the rest is lifestyle.
And if we compare the pharmacological and non-pharmacological approaches,
Well, I don't have a lot of good news for you pharmacologically. I mean, we have some other things coming, but if you look, you know, there was a study a long time ago, and I can't remember who did it now, that compared exercise to Aricept and said which has a bigger effect, and exercise won by a mile compared to our effect in Alzheimer's patients. You know, it's never too late. There's plenty of research also that says if an Alzheimer's patient moves and you get them active and you get them working, you see a slowing of cognitive decline. So lifestyle is, you know, it really is the key to all this. But how do you tell a drug company that? And how do you tell an imaging company that? That's the problem. And, you know, NIH now kind of approves it. But, you know, NIH spent a lot of money on a large trial looking at lifestyle change. But for goodness sake, they did one, you know, we're going to teach you cognition of attention, cognition of executive, cognition, you know, well, it didn't have any effect. It's like trying to study the Mediterranean diet. You got to change the whole beast before you're going to get any movement. And I can't tell you the intricacies of how they're interacting, but it works, you know. The studies of people, are any of you familiar with blue zones? the areas, the sort of names some they were given as sort of areas where people live a long time. We were talking about that earlier today. In Okinawa, where women, unfortunately, my friend who does research there, those women are now getting to be in their hundreds and they're passing away, but women there live longer than anywhere else on the planet. And what's one thing differently they do is they all come together for 2 hours of conversation. They all have tea and they sit for 2 hours and just talk about nice things. You know, we were talking about the Sardinian grape. Sardinia has a grape that people feel is a much thicker wine with a lot of wineskin still in it, but that's where your resveratrol, all those other things are sitting in those grape skins, and that's why maybe those folks live longer. I won't go into the Greeks in the monastery where they eat all the beans. I can't, I'm not sure what effect that has, but they live longer too. So there's a lot of these studies. In California, there's a large group of superagers who their difference seems to be exercise. They're out on the water, they're out doing water tricks and all these other things till they live to be 100. So superaging, looking at why that's so important, what's their secret is the $1 million question. And we're part of a large superaging study going on that's looking for people who have passed age 85 but are still cognitively strong. Why is that? Why is that a lot of those people who are over age 85 have a brain full of amyloid and have no problems at all? What is it special about them? About 20% of the population has a brain that pretty much looks like an Alzheimer's brain, and they go to their grave perfectly happy. You could say, well, they're going to develop Alzheimer's, but they die when they're 90 and 95 and there's no problem. So some people have natural defenses, ApoE3 genes or whatever, that are protective. We just don't know that because, look, if you figure we really started studying this thing in 1985, you can understand why we haven't gotten that far yet.